Good afternoon, everyone. Welcome to part 2 of WCASA's Medical Advocacy webinar series. Today's topic is entitled DNA Essentials for Advocates and Survivors. And we are honored to have Eva King, who's a Forensic Scientist Supervisor at the Wisconsin State Crime Laboratory in the Milwaukee office, and Dan Campbell, who's a Forensic Scientist Supervisor with the State Crime Lab in the Madison office. Today's topic, as I mentioned, is DNA Essentials for Advocates and Survivors, and the issue of rape kits and processing of rape kits and sexual assault kits is something that has attracted a lot of media attention in the recent years, and something WCASA has been working on in collaboration with our partners at DOJ on the Attorney General Sexual Assault Response Team. And I know, speaking for myself, have learned a lot from Dan and Eva participating on the AG Start. So I think this is a really great opportunity for advocates to understand from the crime lab perspective what happens to kids when they are received at the crime lab. And hopefully you find this information useful in your work with survivors, because I know there were things that I didn't understand until I participated in those meetings. So without further ado, I'm going to turn it over to Eva and Dan.
Thank you, Ian. I'm Dan Campbell, and I'm a supervisor in DNA at the crime lab here in Madison. My background is I've been in forensic science now for 24 years. First 17 years of my career was on the bench doing DNA analysis on evidence submitted by law enforcement agencies, and then the remaining 7 years as a supervisor in the DNA unit.
And I'm Eva Marie Lewis-Kane. I work at the Milwaukee laboratory. There I work currently as a forensic scientist supervisor in DNA, but I've been all across the board, all across the state, in fact. I've worked as an acting laboratory manager at the Wausau laboratory, as well as the Milwaukee laboratory. I've also worked as a forensic scientist supervisor in chemistry as well. Started out as a controlled substances analyst, and also worked as a lead worker in toxicology for several years. Currently, as a forensic scientist supervisor, I supervise many DNA analysts, and the crime laboratory, it fits definitely into that part of that do something, take action practice that truly supports sexual assault survivors by delving into the scientific investigation of the sexual assault kit. So I'll start out with Wisconsin statute. In 2011, we received information that we have a new law which reads, whenever a law enforcement agency collects, in case of alleged or suspected sexual assault, evidence upon which DNA analysis can be performed, and the person who committed the alleged or suspected sexual assault has not been identified, the agency shall follow the procedures specified in statute according to the rules of the Department of Justice, that's 165.7778, and shall, in a timely manner, submit the evidence it collects to a crime laboratory as identified in the definition of crime laboratories in statute.
The Wisconsin DOJ SACI campaign for the state has been a huge project currently underway. We'll kind of break through the steps, which essentially are 6 different phases. You'll see on the left of the slide, inventory through report. Initially, what the process started and began with an inventory request from law enforcement across the state, which was estimated to have about 6,000 sexual assault kits in storage at police agencies around the state, at 556 law enforcement agencies statewide. The law enforcement agencies were reached out individually and asked to log and inventory their kits onto a spreadsheet. Each of those kits and the reports associated with each kit were reviewed by a panel. DOJ determined whether to test the kit on several factors, including whether the victim desired to have the kit tested in the first place. Some of the kits, DOJ is still trying to determine victims' wishes and whether to decide to test or not test the kit. To connect kits with victims, it launched a public awareness campaign along with the Wisconsin Coalition Against Sexual Assault. DOJ collected spreadsheets of kit inventories from each law enforcement agency in the state of Wisconsin. The second part was certification. The Bureau of Justice Affairs, which awarded DOJ grant money to test the kits, certified the inventory cataloged on the spreadsheets. This was a requirement before the state received the money to physically send kits in for testing or out for testing. Intake, BJA certified list is then sent to the crime lab, and we act as the central intake point for all of the kits, even though most of the untested kits are sent to a private lab for testing. The crime lab then sends formal notices out to law enforcement agencies asking them to physically send their kits designated for testing to the crime lab. Law enforcement agencies receive DOJ's letter to send the kits, and the crime lab receives the kits, assigns them numbers to each piece of evidence so that we can track that evidence. The fourth step is testing. So the crime lab is sending most of our kits to Bodie Cellmark in Virginia, who will conduct the testing. Bodie Cellmark is an accredited laboratory. The fifth step is review, so all of those results will come back to the crime lab. The crime lab will then review the tests and then eventually report out the results of the testing to each of the law enforcement agencies that submitted the kits.
Now, our aim today is to provide essential DNA information, and we'll attempt to accomplish this by giving you an introduction of DNA. We'll speak to recovery of DNA as presented in the sexual assault kit. We'll discuss the standard crime laboratory testing workflow. We'll mention the processing of sexual assault kits in Wisconsin a little further and go over crime lab report language, their meaning, and also discuss coding. We will also go over specifics on the crime lab's role in the Wisconsin Sexual Assault Kit Initiative, and if time permits, we will briefly go over a real case example. Now to start, we introduce you to the premise and foundation of scientific forensic examination and analysis, Lekars exchange principle. This principle, introduced by Edmund Lekars, states that when two items come into contact with one another, there is always an exchange of substance from one item to another, or from one person to another, or from one item to a person. So this is our foundation that we work from at the crime laboratory, and especially in sexual assault cases.
The sexual assault kit, actually part of the SAKI project and the Attorney General Sexual Assault Response Team, was an effort to revise the sexual assault kits. Historically, they've been, basically the contents of the kit were prescribed by the crime laboratories. There's a medical forensic subgroup that was generated through the AG SART and Wisconsin DOJ SAKI work, where we had a cooperative working group of SANE nurses from the state as well as forensic scientists from the crime laboratory. The kit was revised to help meet the needs in national SANE nursing protocols as well as our needs at the crime laboratory for forensic evidence collection. The kits, part of the project was to offer the kits at no cost to healthcare facilities in the state of Wisconsin to allow them to use the same kit at any healthcare facility. That also assists in training so that it's consistent with all of the new SANE nurses that come on board with the program. The kits are basically sold through the crime laboratory system to healthcare, and then once the kits are collected, law enforcement typically will be picking up those kits from the hospitals and submitting them to the crime lab. Any survivor who has a sexual assault kit collected and doesn't know if their kit was tested for the presence of DNA evidence can call 1-800-446-6564 or go to the BuyYourSideWisconsin.org website to initiate the process of locating their kits and exploring options for DNA testing. Survivors will also be connected with support services.
There's no way getting around it. We have to delve into molecular biology here at this webinar. DNA, it stands for deoxyribonucleic acid. Now, DNA is the chemical for the genome, and it's the genetic code and building block of life. It is a genetic blueprint or instruction manual that directs cellular development. DNA defines things like what you look like at the cellular level. It is a blueprint to build an organism, and DNA stores genetic information and selectively uses the information at the appropriate time. The stored information in DNA is precisely reproduced. And DNA is the same throughout your body, and it's found in all body fluids. It is constant throughout your lifetime. DNA looks pretty complex. However, it is really only made up of 4 nucleotides. We see them as letters, A, G, C, and T. They link things together like the rungs of a ladder, as you see there in the slide. Now, there are 23 pairs of chromosomes in a human, 22 autosomal chromosome pairs, and 1 sex-determining chromosome pair. We get half of our DNA from our mother and half from our father. And as I mentioned, we have those 22 autosomal chromosomes. You will hear us talk about autosomal DNA. That's what we refer to for the main type of DNA that we do at the crime laboratory, and we'll go into this a little bit further. Autosomal DNA is found in every nucleated cell, like white blood cells and buccal cells found adjacent to or in the direction of the cheek. Now, a female has 2 X chromosomes, 1 from the mother and 1 from the father. A male has 1 X chromosome from the mother and 1 Y chromosome from the father. Now, Y chromosome DNA is specific to male individuals, paternally inherited and not unique to individuals. They're unrelated. Male individuals may also share some of the same YSTR DNA profiles. Now, mitochondrial DNA, it's found in the mitochondria of the cell. It's inherited from the mother to offspring. It's not unique to an individual, and hundreds of mitochondria are present in each cell, making mitochondrial DNA testing good for analyzing bones, hair shafts, and teeth. Mitochondrial DNA is not performed at the Wisconsin State Crime Laboratory Bureau. Now, the likelihood that 2 individuals, except for identical twins, will have the same DNA profile can be as high as 1 in a billion or greater. A female has 2 X chromosomes, as I stated, and the Y chromosome, as I stated, is also present only in males. Now, the Y chromosome DNA profile is the same in all paternally related male individuals. And because of that, there's a type of DNA that is very important at the Wisconsin State Crime Laboratory that we utilize called Y screening, or YSTR. We'll delve into YSTR a little bit later, but just to speak a little bit more on Y screening. As we said, it is in the paternal bloodline, and it's not unique to the individual. However, it can be detected from a vasectomized male. It allows for DNA quantification to be used as a screening tool rather than biological fluid screening. So we could go direct to DNA analysis through Y screening rather than doing serological testing, which we'll talk about a little bit later. Y screening is also used for samples where there is a low amount of male DNA in relation to total DNA. And it's better at detecting male DNA from vaginal swabs, external genitalia, and dried secretion. Y screening also reduces the turnaround time. It saves us time on having to screen things serologically, for example, bed sheets and lots of large items. We can go direct to DNA when we perform Y screening at the crime laboratory. And as I said, Y chromosome DNA is performed at the Wisconsin State Crime Laboratory Bureau. Now, finally, I'll talk about nuclear DNA. Now, DNA is found in the nucleus of the cell. This type of DNA is generally the standard form of DNA that you will see or hear talked about on TV, and it's the general form that people refer to when they simply say DNA, that is nuclear DNA. It's unique to the individual except identical siblings, as I mentioned earlier. And nuclear DNA is performed at the Wisconsin State Crime Laboratory Bureau using short tandem repeat, which is we refer to STR analysis.
So we'll talk a little bit about DNA recovery from sexual assault cases. Essentially, Eva mentioned some of the techniques. I mean, YSTR screening has been pretty revolutionary, I think, for the crime labs since 2006, 2007, because it can detect a very small amount of male DNA in an abundance of female DNA. So for sexual assault cases, that can quite often be the case. But sexual assault cases, we have question evidence that may be at a crime scene or associated with the survivor, associated with sexual assault kit evidence is collected at the SANE exam. Suspects may be eliminated based on their evidence profiles, as well as cases themselves can be linked. So we have case-to-case linkages, and that can provide information regarding unknown suspects. We'll talk a little later about CODIS and the database of offenders in the state and as well as nationally, but we can also get hits on cases to evidentiary profiles from sexual assault cases that the law enforcement agency may have no idea who committed the crime or the survivor have any idea who committed the crime. So unknown suspects can also be linked to cases through the data banks work. And our DNA profiles can't reveal anything to us about physical characteristics beyond gender. We have the ability to know whether the DNA profile came from a male or female individual, but it tells us nothing beyond that. We don't know anything about the race, ethnicity, or predisposition to disease through the DNA testing that we're doing at the State Crime Laboratory. Some different types of biological evidence that we commonly encounter and we can recover DNA from. So we have all the body fluids such as blood, semen, and saliva typically are going to have a lot of DNA. Hair with pulled root material with skin tags associated with that root also can have a lot of DNA. Hair that's naturally shed, however, typically does not have a lot of DNA for nuclear DNA testing that we're performing at the lab. So we need that root material or hair that's maybe not naturally shed or forcefully removed. Skin cells also contain DNA, but quite often touch DNA or an item that comes in brief contact with a person's skin for, may not obtain enough cellular material to obtain a DNA profile or get mixtures of DNA from multiple people that may have touched the item. If it's an item that someone has had prolonged contact or handled for an extended period of time, then our opportunity to develop a usable DNA profile becomes much better. If we have mixtures that are complicated enough, one of the limitations of DNA analysis is that we get a mixture that's so complicated or with a number of people in it that it's really uninterpretable or that no conclusive comparisons can be made. Your environmental conditions that can affect DNA are essentially things like heat and humidity, the environment such as water, any type of cleaning products, any bacteria molding, time in and of itself as it passes, DNA degrades. And when it degrades to a certain level, then we are unable to get a usable DNA profile for interpretation. The next table is simply a number of different types of evidence that are relatively routine for coming into the laboratory, things like baseball bats and hats, gloves, and then kind of the location. The middle column is the location where we're typically targeting to either swab or sample for DNA. And then the DNA source in the far right column is just the type of biological material that might be associated with those types of items that we are trying to get DNA from and work up a DNA profile from. We often have very good DNA results from things like a breast swab where saliva is present, cigarette butts left behind at a property crime. those will contain typically an abundance of DNA and are quite often very good items of DNA evidence. Items left behind that are worn by the assailant also, such as a ball cap or gloves, become also very good items of evidence. With sexual assault cases, we have many different types of evidence that might be of interest. There could be blood stains, semen stains, saliva stains, or vaginal secretions or material. Blood stains, sometimes on bedding, weapons, potentially on a suspect that's apprehended by law enforcement, fingernail swabbing and scrapings. Semen stains are obviously one of the more common fluids that we're looking for in a sexual assault case. Condoms, for example, work very well for DNA. Quite often the inside of the condom will contain DNA from the male perpetrator, and then the outer area of the condom containing DNA from the female. Saliva stains, mentioned previously that things like breast swabs as well as the mouth areas of cans and bottles, your saliva contains buccal cells. That saliva becomes a very good material for an abundance of DNA to be left behind. Sometimes we're looking for evidence of a male on female sexual assault. Again, material from the victim that's present on the suspect's body. So things like an external penile swab. Again, condoms can provide that linkage of an item to both individuals at a crime scene. DNA from sexual assault cases, we also can receive items of evidence from hair roots, tissue from underneath fingernail scrapings, clothing from a wearer or something that was left behind at the scene. We've had sunglasses submitted, which also can work very well. Food items, sometimes that seems to me kind of unusual, but we do at times get items of food, particularly things like property crimes too, where someone's coming in and taking a bite of something that's left in the fridge or whatever and leave behind, or decide to open up a can of soda or water and leave it behind, and police collect it and send it to us to the lab. Touch DNA evidence, things like weapons, handles of knives, tools, steering wheels from stolen vehicles. The crime labs will work routinely. We're resourced to work any felony case in the state of Wisconsin, so everything from burglaries up through sexual assaults and homicides and more serious personal crimes. Other types of cases that, other types of evidence from cases that don't necessarily work as well for DNA are things that are listed on the next slide: urine, feces, vomit, and fired casings, and chemically treated tissues. Typically, we have a hard time getting enough DNA and to get a usable DNA profile, interpretable DNA profile from those items. Urine typically does not have enough cellular material to obtain a DNA profile. Feces contains a high amount of bacteria. Vomit, also with the acids and bacteria associated with it, all of those substances can degrade DNA. Fired cartridge casings are something that would maybe be an optimal item in certain cases to examine, but the explosion that ensues with a discharge of a firearm typically destroys and renders the DNA pretty useless for us.
The urine and vomit would be ideal for toxicology at times.
Right. There are many factors. I mean, sperm and semen, the presence of semen is quite often what's of interest in sexual assault, obviously male on female sexual assault. But there are many things that influence whether or not we will be able to detect or find sperm, whether or not ejaculation had taken place, whether a condom was used, whether the person, the suspect has been vasectomized or is aspermic, just the cleanliness and hygiene, bathing, habits of the survivor after the assault, things like bowel movements, the time that's elapsed, the area sampled from the body, and just overall the storage of the evidence. All of those things can have an impact on our success of examining the evidence of the laboratory.
Now, there are limitations of DNA technology. Casual contact generally does not transfer DNA. Oftentimes, we are finding DNA when people have left a generous amount of DNA. Touch DNA, while we are successful at times in finding DNA, oftentimes, 80% of the time, we are not. That doesn't mean contact wasn't made. There just wasn't an exchange of one item to the other. Now, DNA can be removed by washing, and analysis can't be performed in a few hours or overnight, unlike what your television shows would like to tell you. Now, an analyst must be able to visually identify a stain or know where the stain is located on an item. We use visualization techniques at the crime laboratory in order to make that to make those stains visible. DNA cannot be used to determine when a stain was deposited. That's a common question we get in court, when the stain was deposited. And the DNA analyst cannot answer that question. Now, as Dan mentioned earlier, there are environmental factors that limit DNA technology as well, that affect the DNA, such as heat, sunlight, bacteria, and mold, as he mentioned earlier.
So in the Wisconsin Saki process, essentially, again, we had talked about that earlier on one of the slides, but getting the kit tested at the lab has been quite a process. We've got a picture of a diagram there of cases of boxes in a dolly, and that essentially, I know at the crime lab it feels some days like that, where we have loads of sexual assault kits coming in, and then getting them categorized in intake and then sending them off to the private lab. We have not been able to fully start the steps until the grant money was received until 2015. But once we determined the number of kits, as I said, we went through, produced an inventory, certified that inventory, did the intake of all the kits as they've been received, and we continue to receive them from the state, and then test them and review the results and send a report out to the law enforcement agency once the work is done. Determinations made regarding further crime lab in-house work or not on some of the cases. Every report is undergoing a technical review by a qualified DNA analyst that does the same type of work. But like I say, it's similar, but not exactly the same to maybe the active case investigations that are coming in because of some of the age of some of the kits that we're dealing with. Kind of, Eva will be the next slide.
Now, at the crime laboratory, we have a standard procedure for working our cases. We first will screen the evidence. Now, this can be serological testing, detecting biological fluid, or what I mentioned earlier, direct to DNA. We can do Y-DNA on the item or the swab. And then we move on to extraction, where we'll attempt to isolate the DNA from the substrate, whatever we have, the bedding, the underwear, the swab. And we move that on to quantitation, where we attempt to determine the amount of DNA, because this will determine the direction we go with the sample, whether we can move on further for further analysis, or whether or whether we need to concentrate it for DNA or dilute it for the DNA to run further on our system. And then it goes on to amplification, where it's kind of like a Xeroxing, where we copy targeted regions, STR regions, which Dan will talk about further. And we move it on to genetic analysis, where it separates the various STR regions and provides a DNA profile of sorts. So we'll get into data and statistical analysis of that profile, and move on to report writing, where we'll compare and look at our results, provide conclusions, and run statistics. We'll summarize all of that, and then it goes through a series of review. First, the technical review, and then the administrative review of the notes and the report.
I think it's important, too, just for people that maybe don't know, any case that leaves the crime laboratory, any DNA case that's worked, has to be technically reviewed by an independent, qualified DNA analyst to go through all of the notes, data, conclusions, and sign off on the report that they agree with the conclusions of the case working scientist.
So as far as we'll break it down just a little bit here. As far as the biological fluid screening in-house, what happens is that we examine each body swab for foreign DNA. And if no probative evidence is obtained from the intimate items, then items like bedding, clothing, and other external items are examined. We use an inside-out sort of method in regard to sexual assault cases. Now, if there is more than one assailant, then all items are examined and all stains are analyzed in the case. But generally, we're asking our investigators to point out the most probative items to move forth in analysis. Now, we talked about why DNA screen. Now, this is a process that's being used for our outsourced cases, the cases that we send to Bodie. And this is a screen before extraction. A small sample cutting will undergo a fast and crude DNA extraction, and the sample is quantitated with human or male detection methods. In our instance, it's male DNA. Now, we also have the screen after extraction. A larger cutting is taken for DNA analysis. A differential extraction is performed, which Dan will talk about in further detail, and the sample is quantitated with human or male detection methods.
So as far as DNA extraction, we have different techniques that we use. Essentially what's happening in DNA extraction is that we're taking whatever the stain is. The stain may contain various biological fluids. We're going to break down those cells, break open the cells, lyse them, get at the DNA. We're going to purify the DNA, which is removing any things that are not of interest, such as proteins, lipids, carbohydrates, any impurities associated with the sample. And we want to purify, and then we concentrate the DNA that's present in the sample. One of the things that's common in sexual assault evidence, particularly if we have, say, semen present on a vaginal swab in a sexual assault kit, is to do what we call a differential extraction. The slide that you're looking at is essentially that process. We take the picture on the far left is showing or depicting a mixture of sperm and epithelial cells, so the white cells being maybe vaginal epithelial cells, the sperm present with them together. We put those, a sample of that, whether it's a vag swab or a stain from underwear, into a tube, treat it with certain chemicals, and then heat it, and then go ahead and centrifuge it down. Sperm, I know one of our colleagues describes them as the armored trucks of the cellular world. They're pretty resistant. In this case, what we've hit the stain with is not a harsh enough an environment to break open the sperm cells. So we'll centrifuge them down and spin the sperm into a pellet in the bottom of the tube. The vaginal epithelial cells, however, in this example, would break open and be lysed, so we can extract off that top aqueous layer that would contain your DNA predominantly from your vaginal epithelial cell donor into one fraction known as a non-sperm fraction or an epithelial cell fraction. The sperm pellet then is washed and then hit with additional chemicals that provide a harsh enough environment to break open those sperm cells. And we also go through and wash and purify. So a stain similar to a substrate like a vaginal swab of sperm cells will become two different samples, an epithelial cell enriched fraction and a sperm enriched fraction. That gives us the ability to try to look specifically at that sperm DNA and who that donor DNA of the sperm could be. Quantitation is roughly put just an effort for us to see how much human DNA do we have. We have the ability with our testing system to also determine how much male DNA is present. A large enough amount of male DNA will give us the ability to move into the autosomal DNA testing method, which Eva described earlier. If we have a very small amount of male DNA in relationship to the overall female DNA present in the sample, that dictates that we move that sample onto the YSTR method, mainly because the large amount of female DNA quenches the male DNA so that the system just doesn't see it, and that's oftentimes what we're interested in. This is just an example, the next slide, on different types of fluids and the amount of DNA that are present in those different fluids. You can see things like blood and semen having a very large amount of DNA. A buccal swab has a large amount of DNA, thousands of nanograms of DNA per milliliter in a volume. Urine, on the other hand, in a volume per milliliter is 1 to 20 nanograms. Although often the urine we're being asked whether we can analyze it are things more like urine stains or urine left on the ground. Urine and snow, it becomes diluted, and it's hard to obtain enough cellular material to get a DNA profile. Amplification, simply put, is just copying the DNA. The photograph of the instrument in the lower right-hand corner of the slide is a thermal cycler. Essentially, we take the components needed to copy your DNA. We can take a very small amount of DNA and then copy the areas that we're interested in, the different genetic markers that we're looking to obtain for CODIS and that we're interested in. It's similar to thinking of all of your DNA, the genome of your DNA being a book, and we're specifically copying certain pages of that book over and over. That's what gives the type of testing that we do, which is also known as PCR, or polymerase chain reaction, the sensitivity that it has, so we can take a very, very small amount of DNA and then be able to amplify it and get a result and obtain some probative information. It's also kind of polymerase chain reaction. Another term might be molecular xeroxing. But we're using kind of a natural replicating process that goes on in our bodies every day with new cellular growth, and we're kind of replicating that within a test tube by adding the right reagents and chemicals that we need to create an abundant amount of DNA from a very small sample. So PCR has kind of revolutionized the forensic DNA process in crime laboratories.
Another revolutionary process at the crime laboratory, or technique rather, is the use of robotics in the DNA laboratory. Now, these are, it's not a Lexus. You can't tell it what to do, and it'll just go ahead and do what you need it to do. And it's not the Jetsons either. However, it is a liquid handling system that uses disposable tips and it pipettes samples. It aspirates samples and it dispenses liquid for sample mixing. And it does the preparation for further DNA analysis. So that alleviates the analyst from having to do that manual pipetting at times. And we have several different types of robotics in our laboratory. Currently, you're looking at a picture of the TCAN Freedom Evo 100. We have 100 and 150s in our crime laboratory, in our DNA labs today. And they help complete the extraction process by concentrating and purifying the DNA samples. And they assist in setting up the quantitation assay. They assist in normalizing the DNA samples to get the target amount for amplification, because you want that target amount. You don't want too much or too little. And it assists in setting up the amplification assay as well. We have other robotic procedures in-house, and actually in the process of setting up even more right now. Currently, we have that the Eppendorf EP Motion 5070 in place as well, and that's used to set up daughter plates for runs on the genetic analyzer. So we have lots going on. Currently, we're setting up some new extraction procedures robotically. Now, just a word of caution regarding the DNA analytical process. We can't complete DNA analysis in a few hours. That just can't happen. This isn't, as I said before, it's not TV. It takes time to complete the DNA analytical process. We need at least a minimum of 7 hours just to process the evidence samples. That's just the initial stages of it using our current kits and instrumentation. And this doesn't include the time for screening and the data interpretation, the report writing, or the review. And what's not mentioned here are our quality assurance processes that we also have in place that may also need to be applied to each particular case. So it takes time to do a DNA test. And for sexual assault and homicides, those tend to be the larger type cases and are more time consuming, whereas I have seen sexual assault cases go as long as 60 days, sometimes even 90 days. So it can be very timely. And when you put that on top of multiple cases coming into the crime laboratory with limited resources of staffing, it can be a time-consuming process.
We'll talk a little bit about short tandem repeats and STRs. You'll hear that terminology used along with PCR analysis. STRs is a type of PCR testing that crime labs around the country and world are using. Basically, it's that nuclear DNA test. Our YSTRs are also an STR type of testing, where we're looking at short pieces of your DNA that's repeated a specific number of times at a particular genetic location on the molecule of DNA. The number of repeats are what we use to determine an allelic value or a number of the genetic profile, which you'll see a few slides later. Again, YSTRs is targeting specifically male DNA on that Y chromosome, which is exclusive to males. They provide some clarity when there's little male DNA present or an abundance of female DNA present in the sample. So up on the current PowerPoint slide, there's an example of a short tandem repeat at just one marker. We talked, or Eva spoke before, how half of your DNA is inherited from your mother and half is from your father. This one region of DNA and one marker that we're interested in, one of the repeating units on one strand of the DNA has 7 repeats. We would call that type a 7 at that marker. The other strand has 8 repeats, and we'll call that an 8 type. So this individual would be a 7,8 at that location. We're testing over 27, I believe, locations currently simultaneously in one reaction. They call it megaplexing. So a lot is going on in a tube when we're amplifying it. Again, just a small amount of sample is necessary for STRs, and it's highly variable between humans. Most of our DNA is the same within an individual, but we're obviously interested in these variable regions because we want to be able to distinguish between people and identify folks. The STR typing that we're doing in those 20 plus locations that we're testing include the 20 core CODIS loci, which we'll talk about. That allows for data sharing and what allows CODIS to work. We all have to be comparing apples to apples. So we need to be typing the same set of markers to do those comparisons. And then we also use STRs in paternity as well as human identification, things like September 11th and the trade centers, STRs and YSTRs and those types of testing that we're using for forensic analysis. In case investigations, we're also utilized to help identify remains to get those back to folks and their loved ones.
So there are many differences between individuals. However, you may be surprised to know that much of the DNA present in individuals is pretty much the same. However, we're very interested in that small amount that's actually different that causes the differences between individuals. And we call that junk DNA, actually. That's what many people refer to it as, the junk DNA. That's what we're actually looking at, that small percentage of differences between the individuals that allow for the different loci to have the different numbers that Dan mentioned earlier. So we can see characteristics, different, we can see the gender, we can, but we have to make sure that we remember that we cannot, for the type of analysis that we perform, we cannot see physical characteristics beyond that gender, as well as race, ethnicity, and predisposition to disease in forensic analysis. Now, one of the advantages of STR testing, it can be performed on an extremely small amount of sample, very sensitive. It can be done on degraded samples, those degraded samples that Dan talked about. We've had some success with degraded samples as well. and it only uses a small amount of sample. It's fairly rapid, and it's highly discriminating between individuals. So we do use this as a form of identity, a statistical identity, but we use it as a form of identity. And some of the disadvantages of STR testing is that it truly is susceptible to contamination. It has the strength of potentially shutting down an entire laboratory if contamination is introduced. It still requires more than a single cell, and mixtures are very common, especially due to the sensitivity. Interpretation can be very difficult or impossible, so it's very time-consuming on the back end for DNA analysts to sit and look at mixture and interpret those profiles. And it's extremely expensive as well. Now, in January of 2017, the FBI required every laboratory contributing to the national database to expand the number of combined DNA index system, that's CODIS, core loci in the United States from the mandatory 13 CODIS loci to 20 short tandem repeat STR loci, to increase international compatibility for data sharing, to increase discrimination power in missing persons cases, and to reduce the potential of adventitious matches from becoming greater within the data set due to the increased profiles being added to the National DNA Index system annually. Now, the FBI provided labs across the country ample time to make the transition. Most labs use commercial kits for genetic analysis, using multi-megaplex kits, which Dan earlier mentioned, to receive the increased discriminating power and the other improvements in the sample analysis. And the Wisconsin State Crime Laboratory successfully transitioned to the different kit this past January.
The next slide is going to show basically a DNA profile. What we're really looking at, what we're uploading into CODIS for searching, is a pattern of numbers. That pattern of numbers becomes quite unique over all of the loci that we're looking at, except in the case of identical twins. Obviously, people that are related will have a greater likelihood to share different allele types at their markers, but again, even with siblings and parent-child relationships, we're able to discriminate between those individuals with the type of DNA that we're doing. We isolate DNA from the evidence in that series of numbers, and on the far left column it'll say locus. Locus is just the genetic marker that we're looking at. We've talked a little bit about gender marker. You can see the first locus in the table is amyogenin, which has an X and a Y chromosome, which tells us that this profile is from a male individual. The combination of those numbers is similar to a barcode and statistically unique to a person. I know statistically when we look at having the same marker at one location can become maybe not super rare, but when you go across all of those locations, it's kind of like hitting the jackpot on the lottery. It doesn't happen very often. So we have an example now of a nuclear STR DNA profile. That's what a DNA profile looks like. That's a relatively simple profile. It's a single source profile. If you look at each of the markers, they're the bright green on the top screen and the blue dye. Those are the different markers that we're testing at each location. You'll see one or two peaks. In this particular example, most of the peaks across that top blue shade are what we'd say are two alleles, or one from mother and one from the father, and those types being different. That's a heterozygous type. If there is a single allele, we'll say that is a homozygous type. The type of chemistry they're using is 6 different dyes. And again, what we're striving for is something that's statistically pretty darn unique to a particular person.
And if there's a single peak there, that doesn't necessarily mean there's just one contributor there. There's a contribution from the mother and the father at the same location.
The same type.
Allele type.
It's been inherited, you bet. The next slide is of a mixture, which becomes more complicated. Essentially what we do is count peaks. So at one of the markers, if we're seeing more than 2 peaks, and we're seeing that across the profile as we are here in many of the locations, we know that we're dealing with a mixture of DNA. from a person, from more than one person. We can see mixtures of 2 or more people, 3 or more people, 4 or more people. The more people we throw into that mixture, the less conclusive we can say about the DNA mixture with comparisons to someone. Statistically, an inclusion becomes less and less. The mixture gets messy enough at some point, it becomes essentially uninterpretable, and no comparisons are made. The next slide is a YSTR profile. This again is that type of testing that's specific to a male individual. Because we're testing just a marker on the Y chromosome, you're seeing a series of single peaks. What's interesting about the Y profile is the Y chromosome is inherited unchanged from a father to a son. So along that paternal lineage, my DNA profile is the same as my brother's DNA profile, is the same as my dad's YSTR profile. That becomes useful in some cases for us. It's not as discriminating as the type of autosomal standard STR typing that we're using for CODIS, but it does provide us some valuable information where maybe that particular tool is not useful because of the amount of female DNA. It gives us some information to that small amount of male DNA being present.
So earlier I talked about the amount of time it takes to analyze a DNA kit or a case. Now, in the data analysis process, the evidence samples are always analyzed before the known standards. This definitely reduces the possibility of any bias. We examine data in order to determine the profile quality, to determine the number of contributors, which Dan spoke of, and we sometimes attempt to deduce profiles, either a major profile, a minor profile. Sometimes we see trace profiles, and we call some foreign profiles as well, so, or foreign alleles or present. Now, some of the phrases that we use to describe the profile often seen in the crime laboratory reports are single source profiles. This is DNA from one contributor. This is generally recognized by the good quality presence of no more than 2 alleles at each location tested, as Dan mentioned. We have the mixture profile where there's DNA from more than one contributor. This is generally recognized by the presence of 3 or more alleles at each location tested and or peak height balance. We have partial profiles also, and in these, information is generally missing from some or most of the location types. And sometimes we simply have no results. No DNA was detected at any of the locations. Now, it's important to collect and analyze the anticipated single-source survivor's profile at a separate space and time from the evidence samples to establish chain of custody and deduce out a male component if present. So in our data analysis, known samples processed at a separate space and time from those evidence samples is extremely important, and we make sure that happens at the crime laboratory. Now, elimination samples such as consensual partners, they're often submitted for comparison, and oftentimes crime laboratory analysts are calling law enforcement agents to receive those if they were not submitted in the crime laboratory kit or with the crime laboratory evidence samples submitted to the crime laboratory.
The next slide is basically, again, a DNA profile and then frequencies or statistical calculations that are made for that DNA profile. If you look, essentially we have a series of numbers at each of the markers tested, depending on what population groups. Some types are more prevalent in certain racial groups. We can't identify the race based on whatever the profile is most common, and that doesn't hold true. But you'll look to the far right column and you see some of these crazy numbers that we get now where we have in the Caucasian population, this DNA profile that we're looking at occurs and be expected to occur if we're randomly selecting from a population of Caucasians. 1 in 15 octillion would be the number. That's a 15 followed by 27 zeros. We have a policy at the crime lab where we use source attribution to attribute source. If the DNA profile is rarer than 1 in 7 trillion people, we say that this person is the source of that DNA. Most of the numbers that we're getting statistically for the full DNA profile far exceed that number 1,000 times the population of the world. Our DNA reports have several components to them that we're issuing. Typically, these are going to go right to the law enforcement agency that submitted the case and is investigating the case. Our report will have our title, name and address of the lab, case identifiers for each of the items that we've examined. We'll have the submitting agency's address on the report and identifying them. We'll describe the evidence that we received and examined, any significant dates that are needed. The technology has to be described. The results and conclusions are provided in our interpretation. We have both quantitative and qualitative interpretation statements. The date of issuance of the report and the disposition of the evidence is also provided in the report. Signatures and titles of the analyst that performed it, the technical reviewer, as well as the administrative reviewer are also placed onto the report. A lot of the report features that we have to have in a report are required by our accreditation standards to be an accredited laboratory and DNA program.
And because Bodie is also an accredited laboratory, as well as the Wisconsin State Crime Laboratory, you'll see similar features in both of the laboratory reports.
Correct. We have different conclusions that we can arrive at. We can have inclusions, exclusions, inconclusive, no comparisons, and source attribution. I mentioned source attribution. So our statistical method that we use is a random match probability is what it's called. And again, if it's a profile would exist in rarer, would be rarer than 1 in 7 trillion people will say this person is the source of that DNA profile. An inclusion, another way to say that would be you can't be excluded. A match, basically it's one DNA profile from evidence being consistent with the DNA profile from a particular person that we're comparing it to. An exclusion means it does not match, it's not consistent. That person cannot be included as a possible source of that DNA profile when we make the comparison. Inconclusive is just limitations to the DNA profile. Maybe it's a very nasty mixture. Maybe it's just a few peaks are detected, a very partial profile, but we do have some profiles that we say are inconclusive. We can't make an absolute conclusion, either an inclusion or exclusion. And then we also have some that are just not suitable for comparison statements, usually very messy mixtures. As far as our reporting of frequency estimates, we'll say the probability of randomly selecting an unrelated individual with this DNA profile is 1 in whatever in the Caucasian population, and break that down into the different racial groups that are utilized in our statistical program. We generally are going to use Caucasian, Hispanic, and African American. We utilize a population database that's provided through a program known as PopStats that's within our CODIS software that we utilize at the laboratory from the FBI.
So some of the report statement examples that we have at the crime laboratory, quickly read through some of these. A DNA profile was obtained from the sperm fraction of the vaginal swab. The DNA profile obtained from the sperm fraction is consistent with a single contributor. The DNA profile from the epithelial fraction of the vaginal swab is consistent with a mixture of 2 individuals. We've discussed all of those. So the conclusion example here, the possible contribution of DNA from Jane Doe to the STR DNA profile detected from the swab is inconclusive due to the limited amount of genetic information available. So those are some of the type of conclusions or examples you will see in our report. Here are other examples as well. An STR DNA profile that is a mixture of DNA from at least 2 individuals was detected from 1 swab. This mixture includes at least 1 male contributor. Assuming DNA from only 2 individuals, a major female contributor can be determined. The minor component of this mixture profile does not support any comparisons to other STR DNA profiles due to the limited amount of genetic information available. Jane Doe is the source of the major female contributor, STR DNA profile obtained from the swab. Another example is provided here. An STR DNA profile that is a mixture of DNA from at least 2 individuals was detected from one swab. Jane Doe and Janice Doe are included as possible contributors to the STR DNA mixture profile detected. The probability of randomly selecting an unrelated individual that can be included as one of the possible contributors to the observed STR DNA mixture profile is approximately 1 in 8 million. Janet Doe and Jan Doe are excluded as possible contributors to this STR DNA mixture profile. So we've provided you with examples here of the various types of conclusions that we have. And in that first conclusion there at the top, you see in the box there, there was mention of at least one male contributor. Sometimes we can even go on further and do further work, DNA work, on a case in that particular instance with such a conclusion. Sometimes that comes at the request of the agency, and there are times where it's necessary to do it at the laboratory prior to reporting. So, what does all of this mean? DNA is only one piece of the puzzle. What can be said at trial? Generally, we have to testify that there are many possibilities for explaining a DNA result, but how probable are some theories or possibilities? Appropriate significance and relevance of DNA on an item may be helped with scene reconstruction, witness accounts, cell phone data, and etc. All of this to say that DNA is only one piece of the puzzle. The investigation definitely done by the investigator, and there are many pieces to a sexual assault investigation, and as I stated, DNA is only one piece of that.
So we'll talk a little bit about CODIS. I think most people have probably heard the term, but CODIS is just an acronym, stands for Combined DNA Index System. We're a CODIS participating lab, as well as most government crime labs across the country that are accredited also are participating in CODIS. We basically are using the FBI, CODIS is the FBI's program. They allow us the ability to participate as long as we're meeting our accreditation standards in the laboratory. Only accredited laboratories can participate. What CODIS, when we're uploading a profile and we're searching it and we get a match to another sample in CODIS, the FBI and we speak of that as an investigative lead. We don't make the leap, the crime laboratory doesn't make the leap to say that because someone's DNA was left behind at a burglary scene means that they committed the burglary. That provides information to the investigating law enforcement agency to say that this item that was submitted to us from that scene contains DNA from potentially this person. How it got there, why it's there, we don't typically answer that question. There are over 250 labs around the world, 40 different countries that participate in CODIS, which gives it really its remarkable power in helping to provide those investigative leads. The Combined DNA Index is a database of known DNA profiles as well as our casework profiles. So the convicted offenders in Wisconsin since 2000 has been an all-felon state. So anyone convicted of a felony is required to give a DNA profile for entry into CODIS. Arrestees, more recently, April 1st of 2015, we had a law that was enacted in Wisconsin that allowed arrested individuals for certain violent crimes to also be collected, as well as misdemeanors being collected. All of those make up our convicted or arrestee offender pool, which we're entering evidence profiles from casework and searching against those. Obviously, the casework or evidentiary profiles are also being searched within the software at a really rapid pace. And then any of those links that are made case to case or case to offender, basically we're then investigating that and going to report that information to the submitting law enforcement agency. So we talk about CODIS. CODIS has multi-levels. We have photographs on the top of the Milwaukee, city of Milwaukee, and the Milwaukee Crime Laboratory housed there. And then the lower is of Madison, where the Madison Crime Laboratory. We have DNA in both of those two laboratories at the state crime lab. We have, that's what's known as the local level. So there's a local DNA index system at the Milwaukee Crime Laboratory. There's a local DNA system at the Madison Crime Laboratory. We combine those profiles and upload those to the state DNA level, which happens to be housed in Madison, but that contains all of the DNA profiles established in the Milwaukee Crime Lab, the Madison Crime Lab, as well as all of our offenders that have been developed for the state of Wisconsin. That's the state level. And then we upload a couple of times a week to the national level, and there we're being searched. The DNA profiles from Wisconsin are being searched against any of the DNA profiles in CODIS at that national level, which is over 15 million DNA profiles at that national level. We talked a little bit about multiplexes and megaplexes in our kit in CODIS. Eva had mentioned how the requirement of the FBI at the beginning of this year was to have an increase of 20 markers. Those markers are defined by the FBI for government labs that are accredited and are participating in CODIS, that we have to test and make an attempt to obtain DNA from all of those markers. That expanded from 13 prior to the beginning of this year. So how does CODIS assist law enforcement? If you look at the evidence profile on the left, that number pattern is input into CODIS. It's searched against any of the DNA profiles in the system very rapidly. If it finds a matching, consistent DNA profile, it links those two together and reports that back to the crime lab. That match then is what we consider, again, an investigative lead, and we will send a report of that identifying information associated with the offender once confirmed. And then we request a new buccal swab to be submitted from the individual that's matching to confirm that match. We collect that buccal swab or receive it at the crime lab, we analyze it, and then we issue a report to the law enforcement agency. So again, how many profiles are we looking at? You know, and you can look at the different years in each of the rows, 2014, 15, and 16, and you can see the numbers increasing. Obviously in 2014, the middle column, arrestees, we didn't have an arrestee law in Wisconsin, so there was 0. But as we went into 2015 and 2016, you can see the number increasing. I think it's important to note that DNA profiles in CODIS, so from evidentiary profiles, convicted profiles, those profiles stay in CODIS unless legally that we're required to expunge them or remove them. But for the most part, they're going to stay in CODIS, and they'll continue to be searched as new profiles are entered. This gives you an idea of, you know, over 16,000 evidence profiles entered in Wisconsin, you know, in those 3 years that we're looking at currently, as well as the arrestee, and then convicted offender samples, over 230,000. Those numbers continue to grow every day. Wisconsin Act 20 with the misdemeanor convictions and those arrest samples have greatly increased the number of samples that we have in CODIS. The benefits of using CODIS, it can help steer an investigation, you know, quickly. We use it and it's thought of, I think, as a CODIS hit to identify a person. So if we have a case where the victim or survivor or law enforcement may have no idea who committed the crime, it's unknown, a stranger rape, CODIS can become quite a benefit to potentially get a link to a semen profile that's uploaded, and then that information can be quickly given to the law enforcement agency. It can exclude potential suspects. We've had a number of cases where a particular suspect or someone that's of interest to law enforcement is excluded through the DNA, or CODIS hits to someone else in the case. Reduce wrongful arrest, assist in exonerating wrongfully convicted, linking cases within a state and across the nation that maybe law enforcement weren't aware are linked, and it provides some validation to the survivor. I think it's important when we talk about that investigative lead and the CODIS hit, we just don't take CODIS's word for it. We're talking about batches of large numbers of samples that are going into CODIS. those samples maybe don't have the proper chain of evidence or integrity needed for court purposes. So that request of a reference sample being submitted to the laboratory gives us an item of evidence that comes into the lab with the proper chain of custody. It also confirms what CODIS told us, so that we're making sure we, again, reproduce that DNA profile and get the same result for future use in court. What's required to report a match? We use offender fingerprints that are provided with the sample. We confirm the offender's DNA profile. We talked about that process. We verify that the offender, that there was a suitable offense, that the offender owed Wisconsin a DNA sample, and we determine whether or not the crime has already been solved. All of those processes, those checks and balances, again, are making sure that we're not, like as I said, taking CODIS's word for it. We're making sure that a person's DNA sample should be in CODIS, and that we're dealing with the DNA profile of the person that we believe that we are through CODIS, through the HIT. A data bank report example is provided. Basically, we're notifying the agency through this method to let them know that we had a hit to a sample that they had submitted, and it was entered into CODIS and hit on March 17th, 2017 in this example. In this case, we've got agencies, Toontown Police Department as well as Movietown Police Department, and agency contacts that are listed. So we provide that information to agencies on the evidentiary profiles that are linked. In this case, essentially, we have a couple of different cases then being linked together, and items and DNA profiles. The lower line is the Wisconsin offender then that links to those cases. So we're providing information to both submitting agencies that their cases are linked, and then also that those cases are linked to this Wisconsin offender. In this example, Sylvester the cat being linked to the evidence submitted. The next example is a real-life example showing cases are linked. So in October of 2014, we had an armed man follow a woman into her home and sexually assaulting her. And then the crime lab received evidence and went to work. In November of 2014, a male DNA profile was obtained from the labial swabs that were entered into CODIS and searched. In November of 2014, that same time frame, we had a match between the 2014 sexual assault and a 2011 sexual assault that was identified and reported. We have another case example, and Eva and I were discussing cases to put in. I thought this was a case just that I had worked, and we'll speak about it generically, but sometimes we get focused on DNA and body fluids and things like that, and sometimes there's some other interesting types of evidence that if you keep an open mind, you can find some pretty interesting things that might be probative to a case. I had a sexual assault case that I was examining a handful of years ago. And one of the items that was submitted by law enforcement was a comforter. The victim was assaulted in her bed at home. In looking with an alternate light source in the dark, looking for semen stains or saliva stains, anything that might fluoresce, I noticed that the bedding sparkled. There was a lot of glitter associated with the bedding, which probably used some sort of body lotion or powder with glitter in it. One of the things that the police were able to do in their investigation was apprehend a suspect in the case relatively soon afterwards. He had his clothing that was submitted as evidence, and I thought to myself, I wonder if he took any of the glitter from the victim's bedding, since the assault happened at her home and residence, and whether I could find that in the suspect's clothing. So I laid out a piece of examination paper, held up the suspect's jeans, and just started batting them. And debris was falling from the jeans along with pieces of glitter that were falling from the suspect's jeans. Those, obviously we didn't do DNA on the glitter, but I did transfer it to our trace evidence and asked them the question, can you do anything with glitter? And they said actually the shape and thickness and layering process of glitter, that there is something to be said about it. So we searched the internet, even I found an example of glitter can be used as forensic evidence. In this case it was. I had DNA results also in that case. The prosecutor was excited because he felt like jurors would identify maybe with glitter more so than listening to the boring science and DNA results that were present in the case. So it's been a really interesting career. I think that's why Eva and I have hung around for a long time, and satisfying. But anyway, I know, looking to hold questions to the end, and I think we're at the end of the presentation here. So
thank you.
Thank you, everyone, for allowing us to present and fill you in on DNA.
Okay, the first question that's asked of us: can you explain more about the 3 types of profiles that Wisconsin CODIS searches through: conviction versus arrestee versus casework? Yeah,
the conviction and arrestee make up the pool of people, DNA people, or people associated with DNA profiles that we're searching against. Casework being evidence profiles. So if we're developing sperm from a vaginal swab or a male profile identified on a victim's underwear, those are going to be evidence profiles that are associated with casework. We're going to upload DNA profiles from evidence, casework profiles, which may hit 2 other casework or evidence profiles, as well as those convicted certain misdemeanors and arrestee samples that are named to certain violent, usually sexual assault offenses or homicide offenses that someone's arrested for, or convicted of any felony in the state of Wisconsin. Again, we upload to the national system, so we'll search against their profile, so we have their offender pool, depending on what their state laws are in their state and what they can upload to the national system. So we have evidence profiles and then profiles from known people, if that answers the question.
Next question: can you show the slide that said what was required to report a match, please? Sure. Oh, did I pass it already? There we go.
And those are just speaking in general, the different things that we're looking at. When someone gives a DNA sample in the state, they also have to supply their fingerprints associated with that DNA sample, that buccal swab or cheek swab that's collected. That allows us to verify that the fingerprints are from the person that we believe the buccal swab was donated from. So that's a check and balance. Like I say, we're already looking at the DNA profile and requesting that final sample to come back in to confirm that the profile that was in CODIS and matched, is the same. We're verifying that the person that the convicted offender sample that was in CODIS, that person legitimately owed Wisconsin a DNA sample convicted of felony, making sure that they have a suitable crime, that their DNA sample should be in CODIS. All these checks and balances basically to make sure that we're doing what we can do to, like I say, we're not just taking CODIS's word for it. There's a lot of samples in CODIS, so we want to make sure before we release information to the law enforcement agency that we're releasing information about the right person.
Next question. Are elimination samples, profiles, and victim profiles then destroyed, or do they stay in CODIS?
Elimination samples, CODIS, the FBI doesn't allow us to upload any profiles from elimination samples, consensual partners, husbands, boyfriends, the victim themselves. CODIS, early on in my career at the lab, I had been at a conference, and Barry Brown at that time was the ENDIS custodian, or the head of CODIS with the FBI, and he defined it as an index for suspects. So we're looking for individuals or anything that the FBI's terminology is, any DNA profile that could be attributed to the putative perpetrator of the crime. We have a QA index in CODIS that we utilize to monitor for contamination. I think Eva had spoken about contamination concerns within the lab. So obviously any DNA profile or sample that we're dealing with within the lab, we want to monitor that for a certain period of time. We utilize 3 months to essentially make sure that that evidence sample isn't contaminating our work, another sample, another case. And we do that for a period of 3 months, and at that point are comfortable feeling that the DNA wouldn't be have an opportunity to contaminate the evidence.
Did you say the victim's DNA does not get uploaded to CODIS? Wasaki said it did at a conference 2 weeks ago. Is this recent? What Dan spoke of earlier in stating that the victim's DNA does not get uploaded to CODIS, indicating that it doesn't go to national. Locally, he spoke about the Quality Assurance Index. We keep that for contamination purposes. That is where the victim samples will stay for 90 days in order to screen for contamination in the laboratory and have a source for contamination should we have a contamination event. So it is present in our database, but it is not uploaded
into CODIS,
the national level.
One of the things we do, and we have to get consensual partner samples on a sexual assault case, part of the sexual assault kit exam, one of the questions that's important to the crime lab is to know whether the victim of that crime had consensual sex, within a 5-day period. And we want to know that so that we can get a DNA profile from the boyfriend or consensual partner to make sure that any semen identified with the kit is not associated with that boyfriend or husband or consensual partner and uploaded into CODIS. At the local level, again, we're looking at any DNA profile that we have in CODIS to monitor for contamination as part of that Quality Assurance Index. But those profiles are not uploaded to national. It's that national CODIS level that we had the slide depicting.
Hi, everyone. This is Ian, and as one of the Wasaki presenters at a conference 2 weeks ago, Keely and myself, I appreciate the question, and I think that was one of the big things that we talked about at the Attorney General STAR team. Like, if a victim's DNA, where does it go? Does it go to CODIS? And I appreciate the clarification that it doesn't go to CODIS, and there's a possibility it does get. It does go into
the local database.
Local database. So it could be identifying. I mean, it is identifying. It could link to another crime, perhaps. But I really appreciate you both articulating that difference between the local and the CODIS. And so certainly could have been me who confused folks at the Wasaki. I think the point was victim DNA does get run against a local database, but no, it does not go to CODIS. So thank you to Eva and Dan, and thank you to the person who asked the question. Great question.
We utilize it. We have, myself, as an analyst, my DNA profile was in that same quality assurance. We have a staff index at the crime lab. So we want to make sure, as I examine DNA cases at the bench, the person that has the highest potential to contaminate the evidence I'm examining is myself. So if I get a DNA profile and I'm going to upload it into CODIS and think it's a probative value, I want to know that it's not a probative value. It's the analyst examining the evidence. Therefore, it doesn't have probative value, it doesn't belong at CODIS, and it matches me. So that's the purpose of that quality assurance index, is just to monitor for contamination. And like I say, it's not going up to CODIS like an evidence profile, like a cigarette butt from a burglary scene.
It's local. Yeah. It's not in the combined.
So hopefully that clarifies things a bit on that question. It is difficult. It becomes difficult to understand. Sometimes when we're talking about it within the lab or at HSR meetings or SACI meetings that we've had, it takes a while to grasp it. It's not very simplistic, I guess, as far as the levels and all of what's going into CODIS and how long they stay there. Any other questions?
Thank you.
Actually, could you explain a little bit about the difference between a CODIS hit with a named suspect versus a CODIS hit without named suspect? So something that advocates might be seeing coming back as a result of the STR testing, what they're maybe going to need to explain to victims and what that means.
Yeah, we've had, we'll have case-to-case links We've had sexual assaults or sexual assaults that were serial in nature where we've had multiple cases that are linked, but at that time are still not solved if you're looking for the identity of the person whose DNA is linked between all of those cases. We can have a case that has a known suspect that may provide an investigative lead to the agency so that evidence from an unknown assailant case can hit to another crime scene evidentiary profile that didn't have a suspect on the case. And by linking the two, now we know who the person is that contributed it. And we can have, obviously, the hit to a known offender that's in the profile, maybe someone from Wisconsin, maybe someone from Florida, maybe someone from any other state in the country that will provide an investigative lead regarding the identity of who left that DNA profile behind as crime scene evidence.
And those come from convicted offender databases, arrestee databases, as well as what Dan said, other cases as well that have known suspects.
So just because there's a hit to CODIS doesn't mean you're automatically going to know the suspect. You might not know until there's another hit years later.
It depends on the type of hit. If it is a case-to-case hit, and both don't know the suspect, who the suspect is, yes, you could have that type of
Next question, are juvenile offenders profiles entered into CODIS also? And that is dependent, again, the FBI doesn't define for the state what they can put into CODIS. It's dependent upon the state laws that are, so some states may be able to input juvenile offenders, some states not. It just is very dependent on the laws for each state. So yes and no. Yes.
No further questions? Thank you for your time.
Yes, we appreciate the opportunity to talk about DNA a little bit. Hopefully you found it useful and informative.
Contact generally for advocates and survivors is the law enforcement agency due to the confidentiality rules at the Wisconsin State Crime Laboratory. Dan and I, Dan is at the Madison Laboratory and I'm at the Milwaukee Laboratory for just general information. And you can definitely find that in your local directory.
Yeah, the crime labs, one of the things we didn't talk about in the discussion was the crime labs are operated and governed based on what we do and who we communicate, who we take evidence from is defined by state statute. So we have privilege statutes that we have to abide by. So submitting law enforcement agency, the results and analysis and details of the work that the crime lab's doing is privileged to that submitting law enforcement agency. We can't share it with other law enforcement agencies or other attorneys or lay people that are calling victims, suspects. We can't share that, so we share it specifically with law enforcement. That's submitted the case.
That's 165.79.
Correct. Good questions.
Thank you.
Okay. Well, I want to thank Dan and Eva again for presenting today on our webinar. This was, as I mentioned, part 2 of a 3-part medical advocacy series. Just a reminder that part 3 will be coming up on September 21st at 10 a.m. and that will be, the topic will be the Wisconsin Department of Justice SANE, or Sexual Assault Nurse Examiner Program, and the presenter will be Lori Thompson from DOJ. So hopefully you all will register and attend the webinar then. And once again, thank you, Dan and Eva, for your great and informative presentation.
Thank you, Ian.
Thanks, everyone.